Okay, good afternoon. My name is Richard Webb, and this is my colleague Richard Rowlands, so that's confusing already. We're here to cover aspects of providing excellence in laboratory skills and competencies, and how we provide lab-based research projects here in Cardiff Met. Okay, do we have a problem with... There we go. Okay, so like several others of the presentations, I think we were originally invited in 2020 before the pandemic prevented the event from taking place. We were the subject of an accreditation visit in late 2017, about 4.5 years ago, and our 3 courses that you can see mentioned on the slide got accredited at that point. The invitation in 2020 was, struggling slightly with this, there we go, came with 4 commendations. And I'll read them out quickly. The involvement of your technical support team in the education training of students, the thoroughness of the programme's coverage of lab skills and competencies, your work in the further development of lab skills and competencies, and the fact that all students who wish to do so can undertake lab-based projects. A couple of things just to say about those. Number one, I'm really glad that Rich could join me, because clearly you can hear a specific mention of our technical support team, and it seemed totally appropriate for him to take part in the presentation. Secondly, I'm not intending, because of the length of time we've got available to us, to talk about the response to COVID. We're going to kind of take as our brief what the original brief was in 2020 when we were first invited. And thirdly, given the coverage and the discussion there's been on a wide range of different types of research projects or capstone experiences amongst the earlier presentations, the last commendation there, I just want to make clear, the key thing there is who wish to do so. I don't mean to imply that we don't offer a wider range of research projects, but clearly, given the brief we gave, which was based on those commendations, we're going to mostly focus on lab-based projects. I will say, though, that our Student of the Year, who will be at the awards ceremony this evening, did a different type of project. So if you want to hear about the other types of projects that happen in Cardiff Met, you can look for Eleanor Walsh this evening, and she'll tell you all about her project. She'll really thank me for saying that, I'm sure. Okay, so I'm going to hand over to Rich to talk about the next slide.
Yeah.
There we go.
Thank you, Rich. Hi, I'm Richard. I am a technician demonstrator in Cardiff Met. There are around 9 technician demonstrators in total in biomedical science, and we tend to oversee the laboratory-based projects that form a platform for the academic side. Facilities-wise, we have 3 teaching laboratories. One pretty huge one that covers an entire floor, which can hold around 105 students. 2 other smaller labs, which again can work independently, but we have the ability to connect all 3 together to form one big lab. One of the most important parts, obviously, of the lab experience is giving the students confidence, as well as giving them improving their competencies in basic lab skills. That's the idea. So what's good for the demonstrators is that we've got a really good staff-student ratio. On benches of 6, normally in the lab, we can supervise usually up to 18 students per staff member, sometimes even less than that. So the students get a really good teaching experience, which they really do appreciate. So the idea is obviously to make them able to take those skills they learn, especially in the first year, build on them in the second year, and then in the final year, they come again to me, where I manage their third-year projects, and then use all those skills they've developed in those first 2 years to work independently as a project researcher. We've got a designated project lab as well, specifically for third-year projects, which gives them a feel of working in a real lab, and gives them the opportunity to work on their own, as well as within the team of the project students.
Okay, so as I said earlier, we've got 3 courses. Many of you will know Sally Hicks, who's the Programme Director of the large BSc BMS course. I'm slightly more closely associated with one of the other courses, Biomedical Sciences with Health, Exercise and Nutrition. So this is used as an example of the curriculum, as you can see on the slide. A couple of points to highlight are that the little red asterisks indicate modules with practical elements and assessments within them, and it comes out at about 3/4 of our modules take that form. So there's an emphasis throughout the curriculum on practical teaching. There's a sort of modular matrix of the 3 different courses, and you can see the different color codings represent standalone modules for one course, or in some cases, obviously, co-taught modules across the 3 courses. In order to address the commendations that I mentioned earlier, we're going to focus on 2 particular modules, the Year 1 module, Lab Skills and Data Analysis, and the final year Research Project module. The first one, Lab Skills and Data Analysis, relates to the second criterion in the RSB accreditation matrix, acquisition of technical skills, familiarity with the practical environment. And what we do on this module, I think, has links to the coverage we've heard of about skills and competencies from the other presentations earlier today. A pass-fail component of the module assessment are mandatory lab competency tests, or training sessions and tests that take place in the first couple of weeks of the first term. We'll show you some screenshots of the worksheets for those on the next slide, but the idea is, I think there are 5 different tests, 5 different key skills and competencies with regard to, let me get these right, Rich, pipettes, spectrophotometers, balances, microscopes, and microbiological techniques.
That's right.
Did I get them right?
That's right.
Excellent. So all the students will be mandated to attend those sessions under the supervision of either an academic member of staff or a technician demonstrator, demonstrate competency in those techniques, demonstrate that understanding of the theory around those techniques by filling in a worksheet, and be signed off. And if they don't do that, they're not going to get access into the labs for all the later class practicals on this module and on the other modules on the course. So it's a kind of upfront way, as someone mentioned earlier, about making students aware that they've got competencies. Hopefully this does make students aware that, at least in that context of those 5 sets of tasks, they've got competencies. As it happens, I wrote the microscopy one, so here's a quick screenshot. So we've got a tour of the microscope. We've got use of an eyepiece graticle and a straightforward micrometer to produce scale drawings of objects on slides. And we've got, thirdly then, actually been given slides to do that task with, to generate scale drawings with indications of dimensions of objects on the slide. So the whole task has gone through in maybe an hour-long session, and they'll end up with a filled-in version of the worksheet on the right hand of the screen. They've done this, as Rich mentioned, under the supervision of a member of staff, and if they've successfully done all of that, then obviously they're signed off. They get to take a sign-off certificate away with them, and they have the knowledge that they're able to attend the labs for all the other practical sessions later on in the course. So the second criterion in the accreditation matrix, we've taken on board, I hope you agree, the details and the intentions of that, and applied it to our courses. That began in the events around the 2017 accreditation, but we've been continuing with that since. The research project module relates more to, of course, the first criterion in the accreditation matrix, the capstone experience. And you won't be surprised to hear that the most relevant module to that is the research project module. As others have mentioned, the assessments around that, we no longer have a one monolithic thesis. We have, as you can see, a literature review, which culminates in a lit review statement with further recommendations for study, which obviously should relate to the research that they're going to do on the main body of the research project. And then towards the other end of the year, an academic paper in the form of a research article in the journal and a poster presentation session, which happened yesterday for our students. So we had a great day yesterday.
Really successful.
And finally, as Rich will talk now, about a portfolio which equates to being a lab book. And the key thing to say about that is our approach is to have that double marked, unlike the other elements which are double marked by 2 academics. That final element on that little list there is double marked by an academic and the relevant technician who provided the technical support in the research project lab. So you get that, again, that technical focus on that element of the assessment. So I'll pass over to Rich to talk about that aspect.
The final year project is offered to everybody. So we have approximately 100 students, which we all offer wet projects to. Like I said earlier on, we have a designated project lab, so we're able to house them not all at once. We tend to use 2 cohorts, try and get them in at about 50 students each. And it's all run by technician demonstrators. So obviously they get the ideas and they get the meetings with their supervisors. They then come to the lab, and there are 3 of us, 3 technician demonstrators, who oversee all the projects. So obviously we work with the students, give them a sense of ownership of their project. They devise them with us, and then we give them the opportunity to plan their time and their assays accordingly, so they can come and go as they please. Obviously, there was a bit of complexity last year with the restrictions, but we managed still to get everybody through who wanted a wet project. Touching on what Rich said about the competency skills, is something that we try to remind them every time they're in the lab. So although they do it in bulk in the first year, we try to remind them how to do those key skills throughout their course, and especially come the 3rd year project.
Okay, I think somebody mentioned about student projects getting...
sorry.
Publications from student projects, and I'm happily able to say that 4 students who I supervised last year got a publication, as you can see, in exercise medicine. Again, a focus on BMS with health, exercise and nutrition, so the exercise element from a biomedical standpoint, and you can see that we can talk more about this if someone wants to over a coffee, but provisional designation of the interleukin-6 receptor as a novel marker gene for exercise tolerance. People's genotype for that, for polymorphism in that gene, seem to relate to how much physical activity they're able to tolerate, which is an interesting little aspect of human biology nobody had noticed before, I think. So the kind of outcome of that, in addition to passing the module and getting the degree, is that publication. So to conclude, we kind of feel that the science, biology should be a practical thing in terms of hypothesis testing via experimental work, and that's been a big focus of what how we've designed our courses. And there's a couple of NSS scores there, which obviously we score pretty highly there. We've also scored pretty highly in terms of our university's assessment of graduate outcomes data. We really want to finish more by stressing and giving a link for the student blogs page. So Amy Reed, top right in the photos there, a quote from her: "I'd highly recommend studying BMS at Cardiff Met if you want real experience in labs, exciting modules to keep up with current findings, and support from staff that will give you the skills and confidence to start a career as a scientist." Obviously, there are many aspects of that, but we've been asked to talk a little bit about the practical aspects, and that's what we've tried to do.
Thank you very much.
Okay.
Thank you very much for inviting me here to talk about what a small component, actually it's a pretty big component, of what we do in first year practical training at the University of Bath, which is basically looking at how we can integrate lab skills with experimental design in the context of students, first-year students, building their own research projects. So, okay, so why are we interested in doing this? So our experience is that students tend to go to practicals and do what they're told, follow the recipe, and if it works, then they're satisfied, and if it doesn't work, there's not really very much other feedback that they get. This is mostly what they see at A-levels and then, or other, pre-undergraduate experience. And it's actually what they see a lot in their first year training early on. So we wanted to try and address that, which we perceive as a problem. We also wanted to give them a chance to do additional consolidation of their skills, which they don't always get to do. So this is built into many kinds of learning, but not necessarily the same way with practical skills, as we just heard from Cardiff is quite important. We also want to provide them with something that's more meaningful in terms of training them in work conditions. Our students don't often get a lot of appropriate group work opportunities across all the different years, so we wanted to build in some of that as well. And one thing we were really trying to change is that when we had students in their final year, they were really struggling to make these connections between applications, skills, and how to use those to test hypotheses and answer questions. So we wanted to introduce these ideas earlier in their careers. So the context of this is, in our first year course, we have these kind of like different structures. The very first thing that students get when they come in is kind of this boot camp training of technical skills. So these are the kind of things that everybody does, like this is how you use a pipette. Can you demonstrate that you use a pipette? This is how you use a spectrophotometer. Can you do these things? And that's critical to develop those skills, right? Many of our students won't have seen any of these techniques before. But that's just the first step. The next things that we do with them are develop applying those skills in the context of some other research question. So rather than having like individual practicals that deliver a particular topic focus, we have a series of practicals that make up a mini project. So it'll be multi-weeks of answering a big question. We do several different varieties of these, but the idea is that those techniques are all built into a single project that's focused on answering a question. So we've done things like looking at the evolution of drug resistance in E. coli, where students figure out different genetic mechanisms that might underlie resistance. We do some things where we allow flies to make choices about which yeasts they prefer in different contexts, and then measure these outcomes. So they're applying that same series of skills, again, like pipetting, making dilutions, using a spectrophotometer to make measurements, all of those kind of skills, but now in the context of actually addressing specific hypotheses and answering questions that are related, right? So a series of hypotheses that go back to a single question. Most recently, we've tried to develop projects that also encourage student engagement by generating actual research results. So one of my collaborators in this project, we sequence strains that have evolved motility recovery, and students can actually see that the data that they're generating in the lab has real research payoff, potentially. So that's what we do before they start their own projects. So that's what we spend the first semester of our practical unit doing, in preparation for students actually using those skills to design their own experiments. Okay, so they get assigned to groups, so these are typically groups of 6 or so students, and they're gonna work together for an entire semester. So it's really 11 weeks, but it's sort of like about 10 weeks of project work with some extra time built in there. What students get to start is a standard set of things. These are all the skills and most of the materials that they've already seen from the previous practicals in the first semester, and it includes things like their standard elements. So you can have these microbes, you can have this set of equipment, you can have this much plasticware, et cetera. So it sort of like confines them in the scope of the projects that they can design, but they're still being asked to design their own project focused on a question of their own devising. They get quite a lot of guidance in doing this. In fact, every group has at least one group meeting with me or another member of the team who can help them refine their questions, but they're given plenty of time to do background research to figure out if this is a reasonable question. So 3 weeks sort of background before they come up with an actual project design. What they're asked to do in their project design is come up with a formal plan for that project. So they set themselves a question, they set themselves at least one hypothesis that's going to be tested, they design how they'll gather the data to test this hypothesis, and they need to come up with specific plans for what they'll do in the laboratory for each of the weeks that they're allocated. So they're given 4 full laboratory days in subsequent weeks. Depends on the semester schedule, exactly how those are arranged. But they design their own protocols. So there's quite high risk. Students make bad plans. They come up with things that are not possible. So there's a lot of feedback at the stage of experimental design between students and staff. What the culmination of their design is actually them presenting. They make a pitch as if they were pitching a research proposal to a panel. This panel is composed of a demonstrator, staff, team, and they're asked to give a presentation. This presentation is assessed partly on the presentation skills, but mostly on the content of their proposal. This is a two-way process, so students say what they're gonna do, but just like a research proposal, they get feedback from the panel and can make changes as they go. At the end of this pitch session, which happens in 4 weeks in, students are actually asked to say, okay, now you've got some kind of a plan, what do you actually need from us for next week? So students make their own list of required materials in the context of the protocols that they're going to be using. They have to tell us what to give them, otherwise we can't give it to them. They're encouraged to think beyond our standard set of materials. So sometimes they're going to be asking for something extra, like I have to go to the shop to pick up a bunch of strawberries because they want to look at the microbiome of strawberries and how it changes when you add vanillin or something like that. So, but that's their starting point, is to ask for something explicit with a research plan that they're actually going to implement when they come back the following week. Okay. That's the initial process. So they come in with a plan that they've had feedback on that we think is feasible to do in this time period. And then they come into the lab and everything breaks. Typically, there's an immediate impact of students confronting that they don't know what they're doing. There is no recipe to follow except the recipe that they had to write. So usually there's this kind of, that first experience is quite shocking. But at this point, they've now presented their projects and they've been assigned a project manager. So each demonstrator who's part of this assessment team has an opportunity to work with particular groups. They'll work with that single group the whole semester. So they develop this relationship where they know what's going on with the projects. These are PhD students or staff who can provide feedback at all those levels. So students are keeping a lab notebook, which is a good record of their own troubleshooting and response, but they're also getting this constant feedback from demonstrators. So they're having to change these protocols as they go along, including potentially changing their hypotheses. The next step in this process is after they wrap up their experiments, they actually need to confront analyzing the data seriously, so they get some workshops specifically focused on analyzing their data with appropriate statistics. So again, they're getting this feedback to help them do that later on in the process. And in the interest of time, I'm just gonna kind of move on. They get project manager feedback, staff feedback, but they are expected to analyze it seriously. This culminates for the students with a presentation at the end of the semester where they present their entire project, but they're asked not to just present it as if it was a research project. There's also a little bit of the story of the research project, right? So what was the initial plan? How did that plan change? What was the feedback? How did they respond to the feedback? They give these 15-minute presentations, and they are giving them to relatively small groups of students. We have all sorts of things around presentations and how they can do these. These have mostly been oral presentations with slides, and again, to a panel of assessors, so it's a little bit like presenting at a conference. So there are 3 points of assessment in this. There's the initial project pitch, there's the lab notebooks, which are done individually, and then there's presentations at the end. The pitches and the presentations are group-wise. The individual, the notebooks are assessed individually, and critically, there's continuous feedback from the project managers. So, just quickly, in summary, the thing about this project is really that it introduces the students with the freedom and responsibility to try and develop their own work. This really consolidates their skills because they have to figure out how to actually apply the skills in some context. It means that, it's actually quite hard. They get continuous feedback. I think this is valuable, intensive for demonstrators, but it's a good use of our resource. And then finally, this breaks them out of the mold of thinking that skills is all about following a recipe. It's actually about using skills to test a hypothesis or answer a question.